what is an enzyme? We've talked about those. We made all kinds of things about it. An enzyme is a biological molecule that serves as a catalyst for a biochemical reaction. That's it. So it's a molecule in the body, serves as a catalyst. Definition of a catalyst: speeds up a chemical reaction without being used up itself. So after the reaction, it is regenerated. That's the definition of a catalyst. Okay. The majority of enzymes are proteins. Without enzymes to speed up biochemical reactions, life could not exist. We couldn't produce our reactions fast enough to sustain life. So catalysts lower what we call the activation energy, or the energy of activation of a chemical reaction, thereby increasing the rate of the reaction. It basically provides a shortcut. Let's make our reaction. Let's do this. There's our reaction. So if a reaction to occur, this is what we're starting with. This is our reactants. These are our products. Okay. You've got to put this much energy to break all the bonds apart to produce your products, reorganize everything. What an enzyme does, it goes, let's take a shortcut here. Let's go like this and come across right here. Okay. It decreases the amount. This is your activation energy. It decreases the amount of energy it takes for the reaction to occur. So instead of requiring this much energy, it only requires this much energy to go across. It's like taking the shortcut through the mountain rather than going over the top of the mountain. Okay. So this allows the reaction to occur under milder conditions. In the chemistry lab, all we do to speed reactions up is what? Just crank up the heat. Reactions go a lot faster. We can't do that in the body. We can't boil our blood to make it run faster. Doesn't work that way. So the enzyme catalase is one of your enzymes, okay? Provides one of the most spectacular examples of the increase in reaction rates brought about by enzymes. Okay? This enzyme is required for life in an oxygen-containing environment, like what we have. Okay? In this environment, the process of aerobic means oxygen requiring, or oxygen is present, breakdown of food molecules produces hydrogen peroxide. Okay? So breaking down food molecules produce hydrogen peroxide in the presence of oxygen. That's what aerobic means. Our body has some aerobic, some anaerobic reactions too. Because hydrogen peroxide is toxic to the cell, it must be destroyed. If it builds up, you're gonna die. Okay, so one molecule of catalase, okay, that's our enzyme, converts 40 million molecules of hydrogen peroxide to water and oxygen every second. So pretty powerful. Catalase is a pretty powerful enzyme. And it allows it to convert because it's going to be regenerating these over and over and over again. Okay. So one molecule of catalase is going to convert 40 million molecules of hydrogen peroxide to water and oxygen every single second. That's pretty amazing. Okay. Didn't know your body was doing all that, right? So catalase is the enzyme. Hydrogen peroxide builds up. Okay, so you use oxygen to break down food molecules, and it converts it into water and oxygen. Okay, so it breaks down hydrogen peroxide. That's catalase. Nomenclature. How do we name these enzymes? Okay, enzymes are often classified by placing them in categories according to the reaction they catalyze. Oxidoreductase, transferase, hydrolase, lyase, isomerase. Those are different ways of determining what type of reaction is going to be and what the name of the enzyme is going to be that does it. And ligase. Okay. So oxidoreductase, oxidation, reduction reactions. Okay. Oxidoreductases catalyze redox reactions, reduction oxidation reactions. Okay, reductases and oxidases. Reductases catalyze reduction reactions. Oxidases catalyze oxidation reactions. Okay, so lactate goes to pyruvate. It's part of what the cycle we're going to use to break down glucose. Okay, to form ATP in the body. So you take lactate, which is lactic acid that's produced in the body, plus what's called NAD NAD plus. Okay, and we use lactate dehydrogenase, okay, to convert that into pyruvate and NADH. Okay. NADH has been what? This has been reduced. How do you know it's been reduced? You've added hydrogen. Remember, reduction is adding hydrogen, oxidation is adding oxygen. Okay, lactate goes to pyruvate. That is oxidation. You added oxygen. Right there. Okay. So lactate dehydrogenase causes the reduction of NAD plus to NADH and the oxidation of lactate to pyruvate. Transferases transfer a group from one molecule to another. Okay, a methyl group could transfer one group to another group, another molecule. Transaminases catalyze the transfer of amino groups. Okay, transmethylase catalyze the transfer of a methyl group. It's right in the name, transmethylase. ASE means an enzyme. Kinases transfer a phosphate group. Okay. This is norepinephrine converted into epinephrine. Okay. That's being transferred from one group from another. This is, you can see a methyl group has been transferred onto that nitrogen. Okay. So this is, you take any group that's a methyl group donor, and it takes that methyl group and puts it right there and gives a product. And we use the enzyme transaminase to do that. Converts norepinephrine into epinephrine, where epinephrine is adrenaline. And the enzyme we use is PMNT, phenyl, ethanol, amine, and methyl transferase. That's a mouthful, but it's a transferase. It transfers one group from one molecule to another. Glucose goes to glucose 6-phosphate, and it takes energy. We use ATP, and hexokinase is the enzyme we use to do it. And it adds a remember kinase. Kinase means you're adding a phosphate group to another molecule. So we take glucose and ATP and we form glucose 6-phosphate. We've added a phosphate, which is a high energy bond group, to a glucose molecule. Okay. Hydrolases cleave bonds by adding water back in. Hydrolysis, remember? A lot of compounds are formed by dehydration reactions. Dehydration reactions. We move water between two molecules, like a peptide bond. We move H and OH. To put those back in, we can regenerate, rehydrate them by hydrolases. Would cleave that bond, break the bond that we formed by adding water back across those two molecules. Okay, so hydrolases cleave the bond with phosphatases, peptidases, lipases. Okay, those are all enzymes that cause hydrolysis. Glycosidases, like amylase. Okay. Remember our triglycerides, our fat buildup that we have? Okay, remember with the long chain fatty acids, we add water, and we can add water back in between these bonds right here. We add our OH back to produce our alcohol, and then our carboxylic acid. We add H, and then we add OH. H here, OH here. Okay, that's adding water back up. And this is lipase. Triglycerides are fat, lipids are fats. This is lipase. We're breaking it down, we're adding water back across that bond to take triglyceride and form glycerol plus our fatty acid. Lyases catalyze removal groups to form a double bond, or the reverse, break double bonds. Okay, so it catalyzes removal groups that form a double bond or break a double bond. So decarboxylases, okay, synthases. Okay, so you have a few right here. You can see where you have the carbon-carbon double bond. you have your carboxyl gas groups at both tops, and you're going to add water across this bond. Okay, so you're gonna reduce or remove the double bond by adding H and OH. So we're going to attack this double bond, and we're going to add H to this one and OH to this one. Okay, opening up that double bond by adding water to remove the double bond. Okay, and that's fumarase that does that reaction. So fumarate goes to malate. So fumarase breaks down fumarate into malate. Okay. Isomerases. Isomerases catalyze intramolecular rearrangement. So rearrange the molecules in a group. Okay. Epimerases. Mutases. Okay. One example is 3-phosphoglycerate. Go to 2-phosphoglycerate. Okay. The enzyme is phosphoglycerate mutase. So it causes mutation or causes the movement of one group within it to another. Okay? So we've basically moved this group here. We're going to transfer it to, where are we at here? Let me get my bearings right so get in the right place. To right here. Remove that hydrogen. Okay? So that's where we're transferring it. We're moving the group internal within the same molecule. We're just moving that group around. That's an isomerase. Like isomers are non-opposing mirror images we talked about. So we're just moving that group to another place within the same molecule. So phosphoglycerate mutase takes 3-phosphoglycerate and produces 2-phosphoglycerate. Okay. Ligases. Ligase catalytic reaction in which a carbon-carbon single bond, carbon-sulfur single bond, carbon-oxygen single bond, or carbon-nitrogen single bond is either made or broken. Those are your ligases. Okay. And this is a DNA strand. We'll learn the different orders of DNA in a little bit here, but a strand that ends in OH, and then you take a phosphate group. Okay. And a DNA ligase will tie those strands together. Okay. You're making the bond between them. So you're forming this bond right in between these. We're taking water right there. Okay. Allowing this bond to form. So that's DNA ligase. Ligase is counterreaction where you're breaking a bond, okay, or making a bond. Those are your ligases. So to summarize your enzyme classifications, and it's here, maybe oxidoreductase cause they catalyze redox reactions, reduction oxidation reactions. Transferases transfer a group from one molecule to another. Hydrolases break a bond by adding a water molecule back across it. Hydrolase. Lyase breaks a bond by adding a water molecule. Okay. Lyases remove groups to form a double bond or add groups. Okay. So removing those groups to form a double bond, like adding water to it. We add an H and an OH to a double bond to open up to a single bond. Okay. It removes, so lyases remove groups to form a double bond or add groups to break a double bond. Isomerase rearrange groups within a molecule. They take one part of it and put it into another part of the same molecule within itself. And that's an isomerase. And ligase take a single bond, carbon-carbon, carbon-sulfur, carbon-oxygen, carbon-nitrogen, and they either break that bond or form a new one around it. Those are ligases. Okay, so classify the enzyme according to the type of reaction that they catalyze. Okay. So alanine glycine, this is your amino acid, and it breaks the bond. It breaks the peptide linkage right there. Okay. So what kind of enzyme would do that? It would break the bond like that. The reaction occurs here involves breaking a bond, in this case, a peptide bond. This is your peptide bond right here. Okay. By adding a water molecule. You added OH and H. Okay. So the enzyme is classified as a hydrolase, okay, because it adds water across a bond by breaking that bond. This is a peptidase because it breaks the peptide bond. Okay. Okay, in this reaction here, you have the reactions being formed. You're taking this NH3 and moving it to that NH3 group there. You move it to a different molecule. Going from aspartate to alpha ketoglutarate to oxaloacetate to glutamate. Okay? So you're basically transferring an NH3 group. So in this reaction, an amino group was transferred from the amino acid aspartate to the alpha ketoglutarate. So you're going from aspartate to alpha glutamate. You're going from aspartate to alpha ketoglutarate, right there. And then the new product form is glutamate. Okay. So you're producing an oxoacetate and amino acid glutamate. The enzyme is aspartate transaminase. It's an example of a transferase. You're transferring one functional group or one group from one molecule to a different molecule. Okay. Malate, NAD plus, NADH. Okay. So what are we doing in here? You're taking H, going to an oxygen. You're taking this NAD plus and going to NADH. So NADH is being, you're adding hydrogen, it's being reduced. You're adding oxygen, this is being oxidized. Okay, so NADH, NAD plus has been reduced, gaining hydrogen to NADH. Malate has been oxidized to oxaloacetate. Okay, loss of hydrogen, gaining of oxygen. So the enzyme catalysis reaction is malate dehydrogenate. It is an oxidoreductase enzyme called malate dehydrogenase because it causes oxidation and reduction in the reaction. Okay. And dihydroxyacetone phosphate goes to glyceraldehyde 3-phosphate. Okay. And look at the different groups, CH2H with the carbonyl group, and now it's in a different spot here. So groups are rearranged within this molecule. Okay, your carbonyl group has moved to the outer carbon. Both structures have the same number of carbon, hydrogen, and phosphorus atoms. They are structural isomers. Okay, the enzyme is an isomerase. Okay, it's a triose phosphate isomerase. It's moving. Okay, it's rearranging the molecular groups inside the molecule. So it's an isomerase. Because they're both structural isomers of each other. To which class of enzymes does each of the following belong? Pyruvate kinase. That belongs to the transferases. Okay. It transfers one group to another group, one group to another molecule, to another molecule. Okay. Those are your kinases. Alanine transaminase. That is a transferase also. Okay. Transfers amino group between them. Triose phosphate isomerase. That is an isomerase. Pyruvate dehydrogenase. Okay, that's an oxidoreductase enzyme. Lactase is a hydrolase. We add water between it to break down lactose. Phosphofructokinase, again, is a kinase reaction, so it's a transferase. The kinase causes transfer. Lipase is a hydrolase, adds water between them to break that bond apart for peptide linkage. Acetoacetate decarboxylase. It is a lyase. Okay? It breaks the bond between there. And succinate dehydrogenases are oxidoreductases because the oxidation reduction reaction. Okay, so all kinases are transferases. Okay, the isomerases are all isomerase. Okay, dehydrogenase and dehydrogenase are all oxidation reduction enzymes. Lipase, lactase are hydrolases, and kinases are transferases. Okay. So in most cases, enzymes end in the name ASE. They're named the name with ASE. The common name for hydrolase is derived from the substrate. Okay. What it's breaking down. Urea, remove A, replace with A, it's called urease. Okay. It breaks apart urea. Remove the ending A, put ASE. Lactose, remove the OSE and produce N with ASE for lactase. Other enzymes are named for the substrate and the reaction catalyzed. Lactate dehydrogenase. Okay. Pyruvate decarboxylase. Okay. Takes out a carboxylic acid group on pyruvate. Some names are historical. There's no direct relationship to a substrate or any kind of reaction type. It's just what we call them. Okay. Catalase is one. Pepsin. It's just what they've been called over time. Makes it real confusing trying to learn everything. And then chymotrypsin. And you'll learn the common enzymes as we use them. They become apparent. And trypsin. Okay. Those are just historical names that have been assigned to them. So dehydrogenases catalyze the removal of hydrogen atoms from a substrate. So dehydrogenase removes a hydrogen atom. Decarboxylases catalyze removal of a carboxyl group from a substrate. Hydrogenases catalyze the addition of hydrogen. And carboxylases catalyze the addition of carboxyl groups. So you have dehydrogenase and hydrogenase. Dehydrogenase removes hydrogen, hydrogenase adds hydrogen. Decarboxylase removes carboxyl groups, carboxylases add carboxyl groups. Okay, coenzymes. A lot of important reactions in the body rely upon coenzymes. So coenzymes are molecules required by some enzymes to serve as donors or acceptors of electrons, hydrogen atoms, or other functional groups during a chemical reaction. So they work with the enzyme to have their end result affected. So they'll accept electrons, hydrogen atoms, or different functional groups to it, and then allow the chemical reaction to occur. You need the coenzyme to attach to the enzyme for that functional group. So write an equation representing the reaction catalyzed by each of the enzymes. So the first one is pyruvate kinase, which is a transferase. Alanine transaminase, which is a transferase. Triose phosphate isomerase, which is an isomerase. And pyruvate dehydrogenase, which is an oxidoreductase. So the first one is pyruvate kinase. So pyruvate kinase is a transferase reaction. So here's an example of one. It's a group transfer reaction. Pyruvate kinase, you see the phosphate group is transferred, okay, and energy is produced. This is going to pyruvate. This is phosphoenopyruvate going to pyruvate. Okay. See the same molecule? You're transferring a phosphate group onto another compound. Okay, like I said, I can't draw here. So phosphoenopyruvate serves as a donor of the phosphoryl group that is transferred to ADP to produce ATP. Okay, so you're producing ATP in this reaction. You're transferring a phosphate group from ADP to ATP. Okay, this is another coupled reaction which hydrolysis of the phosphoester bond in the phosphoenopyruvate provides energy for the formation of the phosphoanhydride bond of ATP. So the final product of glycolysis is pyruvate. So when we do glycolysis, which we're going to do, I think, either this chapter or the next chapter, in producing ATPs, this is one of the reactions we're going to use to produce pyruvate at the end of what glycolysis turns into. Okay, alanine transferase is a transferase reaction. Okay. You look at the two different ones there. You see L-alanine and alpha ketoglutarate. Okay. And you look at the NH2 group. NH2 group goes from alanine to the glutamate. So that's a transaminase, okay? It's a transferase reaction. Okay, so alanine transaminase catalyzes the transfer of the alpha amino group of alanine to the alpha ketoglutarate and produces pyruvate and glutamate. Okay, you see where the the amino group's been transferred. That's a transferase. And they want to be an alanine transferase, so it's from alanine. So dihydroxy acetophosphatase, this is an isomerization reaction. In other words, if you count the number of carbons, oxygens, and phosphates and hydrogens, it's the exact same on each one. Okay, they're isomers. So this is an isomerase. It's just transfer location is going to be different. Okay, so the reactant and the product each have the same number of carbon, hydrogen, oxygen, phosphorus atoms. Okay, they're structural isomers. Okay, the enzyme must be an isomerase. It's a triose phosphate isomerase is the enzyme. And it's an example of that type of reaction. So isomerase reactions involve moving functional group from one part to another, and it's an isomer of one. They're both isomers. Okay. Pyruvate dehydrogenate, oxidase reductase. Okay, it's a reduction oxidation reaction. So pyruvate goes to acetyl coenzyme A. Okay, and you see NAD plus goes to NADH, and that has been reduced. And the pyruvate, okay, has been oxidized. Okay, so explaining here is the decarboxylation and oxidation of pyruvate. You lost the carboxyl group, okay, and oxidation of pyruvate to produce acetyl coenzyme A. The overall reaction is that CO2 and a hydrogen are removed from pyruvate. Okay. The remaining acetyl group is attached to coenzyme A. Okay. This requires a concerted action of 3 enzymes and 5 coenzymes. So it's a very complex reaction to do it. But it's a pyruvate dehydrogenase. Okay. You've lost hydrogen from pyruvate. Okay. So the pyruvate dehydrogenase complex that carries out this reaction is actually a cluster of enzymes and coenzymes. So it's a group that are doing it. Okay. The substrate is passed from one enzyme to the next as the reaction occurs. So each enzyme is regenerated as it's passed from the next step. So you take the enzyme, it bonds to it, changes it, modifies it, and then something else is passed to the next one. Okay. And you regenerate your original enzyme. So write an equation representing the reactions catalyzed by each of the following enzymes. And again, we'll do lactase, phosphofructokinase, lipase, acetoacetate decarboxylase, and succinate dehydrogenase. And again, this can be any reactions you want. These are just examples of one that I picked on. Okay. So the first one, lactase. Lactase is a hydrolase. Okay. So you have lactose goes to D-galactose and D-glucose. When you look at it, you see the bond that's being broken. Okay. So glucose is liberated by the hydrolysis of lactose. So you're adding water across that dimonomer bond in there. Okay. We talk about, we do the Fisher projection, we do the ring structures. Okay. So your hydrolysis of the lactase bond to G and the energy production reaction of glycolysis. So that's what we do in the ring glycolysis cycle that we run. A bond is broken and water is added between those two. So you break that bond, you're adding H to one side, OH to the other side. Okay, that's our hydrolase. We're adding water. That's what breaks the bond to produce glucose. Okay, phosphofructokinase. Again, we're taking ATP and producing it to ADP. So it's that reverse reaction now. We're using energy to have this accomplished. And we're adding a phosphate group to fructose. To fructose 6-phosphate, we go to fructose 1,6 by phosphate. Okay, so you're adding a phosphate group to fructose. So it's phosphofructokinase, and it's requiring energy to do it. Okay, so the transfer of a phosphoryl group from an ATP molecule to a molecule of fructose 6-phosphate is catalyzed by phosphofructokinase. So you're taking the phosphate group from ATP and adding it to the fructose 6-phosphate. Okay, lipase. Okay, lipase we did with the triglycerides. When we added 3 waters, triglyceride has 3 carbons, and it's a hydrolase as you're adding water across it. So we're going to add an H and an OH, water across that bond to break our fatty acid chain off of triglycerides. We're going to generate glycerol, which is the 3 alcohols, and then the fatty acid chains. Okay. So lipase catalyzes the hydrolysis, adding of water, of the ester bond in the triglyceride. And we break that bond between the fatty acid chain and the alcohol. So we got there by dehydrating between the alcohol and the fatty acid chain. We're reversing it now with lipase, which is a hydrolysis reaction. So it's the addition of a water molecule to a bond resulting in the bond breaking, because we're adding H and then OH to the other side. These reactions are important in the digestive process when we break down triglycerides. Okay, acetoacetate decarboxylase, that's your lyase. Okay, a double bond is added to form CO2 as it breaks off. Okay, so the first step is to remove a hydrogen there, form your carbonyl group, and then we're going to move that carboxylic acid group, okay, and form acetone. We give off a CO2. Okay, and we form the double bond carbonyl group. And that's our decarboxylase. We've lost the CO2. Okay. Succinate dehydrogenase. This is an oxidation reduction reaction. Okay. FAD is going to FADH2, which is then being reduced. It's been adding hydrogen. It's an electron carrier in our glycolysis. So succinate goes to fumarate. Okay. So FAD has been reduced. You've added hydrogen to it. And succinate has been oxidized. It's lost oxygen atoms. Okay. Because you've increased the double bond between 2 carbons as in fumarate. Okay. So what is the substrate of the following enzyme? What is the substrate for each of the following enzymes? So sucrase is going to break down what? Sucrase is going to break down sucrose. Pyruvate decarboxylase is going to break down pyruvate. Takes the carbonyl group off. Succinate dehydrogenate is going to break down succinate. Okay? It takes away hydrogen molecules. You can get that right from the names of those. What chemical reactions mediate to the following? Sucrase is going to break down. Well, what type of reaction? It's a hydrolase. It's going to add water back across it, right between the 2 monomers. Pyruvate decarboxylase, okay, is a lyase. Okay, and succinate dehydrogenase is an oxidoreductase, oxidation reduction reaction. So the effect of enzymes on the activation energy reaction. Remember the activation energy? If the reaction starts here, needs that much energy and produces products here, this is your activation energy. How much energy it takes to get the reaction started. So an enzyme speeds up a reaction by lowering the activation energy. So it changes the reaction pathway. It's like taking a shortcut through the mountain rather than going over the peak. You change the pathway that the reaction is taking. This provides a lower energy route for the conversion of the substrate, which is what you're starting with, to your product, which is your end goal. So every chemical reaction is characterized by an equilibrium constant. KEQ. K stands for constant, EQ stands for equilibrium. It's a reflection of the difference in energy between the reactants and the products. How energy is going to favor either one or the other. It's what a KEQ is. And the definition of an equilibrium constant is the concentration of the products divided by the reactants, each raised to their stoichiometric equivalency. Now, I know that sounds weird, but all it is your balanced chemical equation. These are the numbers used to balance it. You're going to take your products, which is C and D, the concentration of those right here, and you're going to raise them exponentially by their exponent by the balanced numbers, by C and D. So your equilibrium product is your products over your reactants. These are your reactants, and these are your products. So it's products over reactants, and each one of those is raised to the number that was used to balance the equation. That's it. Okay, so here's our equation. This is nitrogen combined with hydrogen to form ammonia. That equation is balanced. There's 2 nitrogens on the left 2 on the right, 6 hydrogens on the left, 6 hydrogens on the right. So our K equilibrium constant is our product, which is NH3. So here's NH3 and it's raised to the second power. Okay, and then hydrogen divided by our products, which is nitrogen, and it's raised to the 1, which is just the same as normal there. It's just the concentration of nitrogen, and then hydrogen raised to the third power. So we can measure the concentrations during the reaction. We measure the product, we know which concentration, we square it, divide it by nitrogen concentration and hydrogen concentration cubed. Okay, that's all the K equilibrium constant is, and we use that a lot. So a diagram of the energy between reactants and products. These are the ones I was showing you here. This is reactants, comes up. This is our activation energy, and these are our products. Okay, when it's catalyzed, we have it here. This is our activation energy, and these are our products. You can see it takes a lot less energy for the reaction to occur. Okay, it's like taking the shortcut, taking the path through the mountain rather than over the mountain. So it decreases what's called the activation energy. So the uncatalyzed reaction has a very large activation energy. And that's this graph on the left right here. Okay. The catalyzed reaction, the activation has been lowered significantly, increasing the rate of the reaction. So if it takes less energy for the reaction to occur, the reaction is going to occur faster. Make sense? Sort of. All right. So no matter how the chemical reaction occurs, whether it takes the uncatalyzed reaction or the catalyzed reaction, okay, the difference in energy between the reactant and the product is always the same. In other words, the energy you're starting with here and the energy you end up with here is going to be the same whether it's catalyzed or not. Because here's the energy you start with, here's the energy you end up with. So if you're not changing the energy between these reactant products, you're just making the reaction go faster. That's all you're doing. Okay, so there's no difference in the energy between reactant and products, whether it's catalyzed or uncatalyzed. It's just making the reaction occur quicker. Okay, so for this reason, an enzyme cannot alter the equilibrium constant for the reaction that it catalyzes. So the KEQ for this reaction is going to be the exact same thing as a KEQ for that reaction. You're not changing the energy or the concentration of the product. They're going to be the exact same for each reaction. It's just a catalyzed reaction is going to occur a lot faster. Okay? Now we can affect the concentration of an enzyme catalyzed reactions, how quick a reaction it goes, depending on what we're starting with. So rates of an uncatalyzed reaction increase as the substrate concentration increases. Okay? So say we have that's going to react and it's going to produce our product. If we take this and we increase that amount of it and we get the same product, this second reaction is going to go a whole lot faster because there's more substrate molecules to run into each other to hit to react with one another. The more you have reacting, the faster the reaction is going to go. Okay, this is not catalyzed, so it's an uncatalyzed reaction. So the rate increases as the substrate concentration increases. Okay? So the rate of an enzyme catalytic reactions shows 2 stages. Okay? The first stage is the formation of an enzyme substrate complex. Okay? The enzyme binds to the substrate. Then it's followed by the conversion to the product. So the rate is limited by the enzyme availability. Okay, no matter how much substrate you have, if you only have a little bit of enzyme, that's all you can run. Okay, so the rate in an enzyme catalyzed reaction is limited by the enzyme. The rate in an uncatalyzed reaction is limited by the substrate. The rate in a catalyzed reaction is limited by the enzyme that's available. Okay, so here's your uncatalyzed reaction. Okay, as you increase the substrate, you increase more of it, you can get a faster reaction. It's a linear scale. Okay. the enzyme catalyzed reaction. Okay, as you increase the amount of substrate, comes up here, and you increase it because there's more to react with the enzymes until your enzyme runs out. Okay, then it reaches the maximum rate. Okay, you can't have it. You run out of the enzyme that's available to bind with the substrate. So those are your different curves. This would be uncatalyzed. This would be enzyme catalyzed reaction. Okay. As you increase the substrate, you increase your reaction rate. As you increase the substrate, you increase your reaction rate up to a point where the enzyme becomes saturated. Then you have a maximum value. There is no saturation point on the uncatalyzed reaction. Okay. The enzyme substrate complex is when the enzyme binds with the substrate. These reversible reaction steps represent the steps in an enzyme catalyzed reaction. Okay. The first step, like we said, involves the formation of an enzyme substrate complex. So the enzyme and the substrate bond together. Okay. This is the enzyme substrate is your transition state. It starts to modify right there. Enzyme product is the enzyme product complex. So you have this enzyme substrate. Let's write right here. So the enzyme binds to your substrate. Then it starts to change. Enzyme substrate starts to modify. And it starts to turn into an enzyme product. And then it's going to separate into the product and releasing your enzyme back. Okay, so when they're bound, they change the cup, they change it. So EP is the enzyme product complex. So look at the different steps. You have your free enzyme. Now remember, for it to be an enzyme, it has to be regenerated at the end. The starting one has to be regenerated. So the enzyme substrate combine to form your enzyme substrate complex. Then there's a transition state where you start changing the substrate into what the product's going to be. And then you still have the enzyme bound to the product once the product is formed. And then the enzyme has to separate and regenerate. And you have your product and your substrate. So those are steps involved in an enzyme substrate complex, an enzyme catalyzed reaction. Okay. Okay. So the part of the enzyme combining with that substrate is what we call the active site. That's the active site of the enzyme. Active site characteristics include pockets or clefts in the surface of the enzyme. Remember, everything works by shape. So if you have a pocket or a cleft where an enzyme can adhere to, that's what it's going to do. Okay. R groups at the active site are called catalytic groups. Those are the ones that help through polar bonds, through hydrogen bonding, through disulfide to salt bridges, like we did with the protein structures. The R groups do the same thing to help hold the active site to the substrate. And these are called catalytic groups. The shape of the active site is complementary to the shape of the substrate. So if the substrate looks like this is your substrate, your enzyme cannot look like that. Those aren't going to fit, right? It's not going to match. So the enzyme, come back here. Okay, this is your substrate. So the enzyme is going to look like this. Where this will fit right in there. Okay, so the shape of the active site, so this is your enzyme, and this right here is your active site of the enzyme. Okay, it's going to have a complementary shape to your substrate. Everything works by shapes to identify it. Okay, so your enzyme has an active site. The active site is going to have a shape similar so the substrate can bond into that area. Okay, and the R groups that surround this active site that cause it to be shaped like that are called catalytic groups. Okay, so the enzyme attracts and holds. Okay, so the enzyme attracts and holds the enzyme using weak non-covalent interaction. Okay, that should say substrate. Not enzyme. Okay, so the enzyme attracts to and holds the substrate using the weak non-covalent interactions, the one between the R groups that we talked about for our protein shapes, for our tertiary shapes, our hydrogen bonds, our hydrophobic interactions, our salt bridges, and there's one more, our sulfide, disulfide bonds. Okay, so that's your enzyme substrate complex. Conformation of the active site determines the specificity of the enzyme. Okay, so when the active site binds to it, it determines that the enzyme specifically fits into that substrate. Lock and key enzyme models. Let's do this and we'll call it a night. In the lock and key model, you can kind of guess. It's going to be like a lock and key. The enzyme is assumed to be the lock, and the substrate is going to be the key that fits into the lock. Okay, the enzyme and substrate are made to fit exactly. This model fails to take into account protein conformational changes to accommodate a substrate molecule, because you can't make conformational changes like isomers. Okay, so here's your enzyme. These are a little bit better pictures than I can draw, but this is a lock and key. So here's the lock, and the substrate is the key. Okay, this is the enzyme. This is the active site right there. Okay, so here's your enzyme. Here's your substrate. Now here's your enzyme substrate complex.